आज हम इम्यूनोलॉजी के एक दूसरे बेसिक कॉन्सेप्ट और बेसिक टर्मिनोलॉजी के बारे में देखेंगे, या कह सकते हैं कि एक दूसरे इम्पोर्टेंट कंपोनेंट के बारे में आज हम लोग देखेंगे। तो ये क्या है? एंटीबॉडी। एंटीबॉडी क्या होता है? एंटीबॉडी को हम लोग इम्यूनोग्लोबुलिन के नाम से भी जानते हैं। ये स्पेशलाइज्ड ग्लाइकोप्रोटीन होता है जो कि प्रोड्यूस्ड बाई एक्टिवेटेड बी सेल्स और प्लाज्मा सेल्स के द्वारा प्रोड्यूस होता है इन रिस्पॉन्स टू एंटीजेनिक स्टिमुलेशन। एंटीजन और इम्यूनोग्लोबुलिन इज ए स्पेशलाइज्ड ग्लाइकोप्रोटीन प्रोड्यूस्ड फ्रॉम एक्टिवेटेड बी सेल और प्लाज्मा सेल इन रिस्पॉन्स टू एंटीजन. It constitutes 20 to 25% of total serum protein. There are 5 classes of antibody: IgG, IgA, IgM, IgD, and IgE. Antibody is Y-shaped heterodimer composed of 4 polypeptide chains, 2 identical light chains which have 25,000 Dalton molecular weight, 2 identical heavy chain which has 50,000 Dalton molecular weight. Each chain have 2 end amino terminal and carboxyl terminal. There are 5 classes of each chain on the basis of which antibody are classified in 5 classes. These H chain are antigenically distinct from each other. These are gamma heavy chain, alpha heavy chain, mu heavy chain, delta heavy chain, and epsilon heavy chain. Light chain are of 2 types, kappa and lambda. Kappa was discovered by Korngold and lambda was discovered by Lipari. 60% of light chain are kappa and 40% lambda. Both the light chain of an antibody molecule are of same type, either kappa or lambda, never both. This means light chain may be either kappa or lambda, never both. Like if any antibody have kappa light chain, then it have only kappa. Other which have lambda only. Here are structure of antibody. This part is from here to here. This is heavy chain and 2 heavy chain, one is here and one is here. Both heavy chain linked through disulfide linkage. This is disulfide linkage, and this one part is light chain, one light chain here and one light chain here. Light chain also connected with heavy chain through disulfide bond. Here also light chain and heavy chain connected through disulfide bond. This is amino terminal and this is carboxylic terminal. This part is variable region. This part is variable region, and this part is antigen binding site or 5 region. And from here, this green parts from here to here constitute constant region represented by FC. Then now we will study in detail about antibody. Light chain composed of 214 amino acid. Heavy chain varies from 446 in case of alpha chain to 476 in mu chain. First 110 amino acid near amino terminal means from here to 110 amino acid. It is called variable region. This is a variable region. This is variable region for light chain VL, and this is variable region for heavy chain VH. Okay. Hypervariable or complementarity determining regions CDR within variable region. In this variable region, there is some region where variability is very high. Is called hypervariable region or parato. Hypervariable region is from parato. There are 3 hotspots in EL light chain and 4 hot spot in heavy chain. Here only. Length of constant region 104 amino acid in case of light chain, and length of this is constant region of light chain. Okay, from here to in case of from here to here in case of heavy chain, it is a constant heavy chain, constant region of heavy chain. Length of constant region is of 330 amino acid in case of gamma, alpha and delta heavy chain, and 440 amino acid in epsilon and mu heavy chain. Light chain contain one variable and one constant domain, and heavy chain contain one variable and 3 or 4 heavy 3 or 4 constant constant domain in heavy chain. Here this is a light chain and it have one variable domain in light chain and one constant domain in light chain. Here one variable in heavy chain, one variable domain in heavy chain, and 3 or 4 constant domain in case of IgG, IgA and IgD, there are immunoglobulin antibody have 3 domain in heavy chain region. In case of IgE and IgM, there are 4 constant domain in heavy chain. What is the function of immunoglobulin? It binds to antibody by 5 region. 5 fragment bear the variable region. 5 fragment bear the variable region is involved in the interaction with the antigen. Valency of an antibody referred to the number of 5 regions it possesses. Monomeric antibody molecule has a valency of 2. Here is a 5 region which bind, this is 5 region which bind with the antigen, and 5 region, and there is 2 valency, one is here for monomeric antibody. Okay, effector function, fixation of complement antibody coating the target cell, bind to complement through its Fc receptor, then complement mediated lysis of target cell, binding to various cell type bearing Fc receptor lead to activation. Example: phagocytes, lymphocytes, platelet, mast cell, NK cell, and eosinophils and basophils. एंटीबॉडी के 2 मेन काम होते हैं, एक तो वो एंटीजन से बाइंड करता है थ्रू एफएवी फ्रेगमेंट, और दूसरा कि एंटीबॉडी जो होता है वो जो फॉरेन बॉडी आती है या एंटीजन आता है, फॉरेन बॉडी लाइक बैक्टीरिया और अदर माइक्रो ऑर्गेनिज्म, या उस पर फॉरेन एंटीबॉडी जाके बाइंड कर जाती है थ्रू, उससे बाइंड कर जाती है थ्रू एफसी रीजन। नो फिक्स एंटीबॉडी जो फॉरेन बॉडी आता है उससे एंटीबॉडी बाइंड करता है थ्रू 5 रीजन। एफसी रिसेप्टर जो उसका फ्री होता है वो कॉम्प्लीमेंट से बाइंड करता है और सेल लाइसिस करता है, बाइंडिंग टू वेरियस सेल टाइप वियरिंग एफसी रिसेप्टर लीड टू एक्टिवेशन। ये प्रमोट करता है फैगोसाइटोसिस, फाइगोसाइट्स बाइंड करके फैगोसाइटोसिस प्रमोट करता है, लिम्फोसाइट्स, प्लेटलेट और मास्ट सेल, एनके सेल, न्यूट्रोफिल्स एंड बेसोफिल्स बाइंड कर। अब हम फंक्शन देखेंगे, मेन फंक्शन ऑफ all classes of immunoglobulin: IgE, IgG, IgM, IgE and IgE। IgD का फंक्शन जो है वो अभी डिस्कवर्ड नहीं हुआ है कि उसके क्या फंक्शन होते हैं। IgG, IgM and IgE have some significant functional role in body, while IgD function is not yet determined or appreciable. What exactly the function of IgD? IgG provide immunity to fetus and newborn as it cross placenta. IgG help in complement fixing. IgG help in phagocytosis, mediate coagulation by reacting with protein A and S of Streptococcus aureus, and IgG is the major immunoglobulin or antibody in case of chronic infection or after recovery of it is a diagnosis or recovery of a patient from infection. IgM, it is antibody formed in acute infection. This is a primary immune response. It also help in complement fixing, most potent in complement fixation, as it is as IgM found in pentameric form. It provide fetus to immunity. IgM is the first antibody which start formation in fetus after 20 week, provide protection against intravascular organism, opsonine and agglutinations. IgE provide local or mucosal immunity. It present in various secretory product of body like milk, tear, saliva. IgE have 2 main functions, mainly type 1 hypersensitivity and helmintic infections. Now we will learn about some important other terminology like isotype. What is isotype? They vary from each other in the amino acid sequence of constant region of their heavy chain. जो IG एंटीबॉडीज़ होते हैं, IgG, IgM, IgD, IgA एंड IgE, ये आइसोटाइप होते हैं क्योंकि इनके जो एमिनो एसिड सीक्वेंस होता है कांस्टेंट रीजन में वो वेरी करता है. Idiotype amino acid sequence present in paratope region of one member of a species act as antigenic determinant to other member of same species is called idiotype. Allotype antigenic determinant present in the isotype gene in the constant region of H and L chain encoded by multiple allele are called H allotype. What are the abnormal antibody or immunoglobulin? First called Vance-Jones protein. Here excess production of light chain seen in multiple myeloma. Waldenstrom's macroglobulinemia seen in B cell lymphoma, producing excess IgM. Heavy chain disease. Here excess production of heavy chain of immunoglobulin that have short and truncated heavy chain. Cryoglobulinemia seen in multiple myeloma and hepatitis C infection. Now we will see what is monoclonal antibody. Monoclonal antibody, it is a clone of B cell stimulated against a single epitope of an antigen. Monoclonal antibody are synthesized by a double mutated myeloma cell when co-culture with spleen B cell in polyethylene glycol broth. Then chances of the formation of three kind of cells: unfused myeloma cells and unfused mouse or splenic B cells and fused hybridoma cell. These cells are our interest cell. Fused or hybridoma cells further propagate for clones, and then for confirmation of for confirmation we test it by antigen to that particular antibody. After confirmation we grow in mass culture and we get monoclonal antibody. Application of monoclonal antibody in diagnostic as a diagnostic reagent, like in ELISA and rapid test. Detection of hepatitis B surface antigen, serogrouping of streptococci. Passive immunity, like in case of tetanus and rabies immunoglobulin, we give this monoclonal antibody. And therapeutic uses of monoclonal antibody in case of rheumatoid arthritis, adalimumab, we use against TNF alpha, use as immunotoxin. Here monoclonal antibody plus monoclonal antibody plus tetanus diphtheria toxin used to kill cancerous cell. Thanks for watching. Do like subscribe my channel and